Please help me welcome Dr. Haik Varsakian.
Thanks, William, and good morning, everyone. Welcome to the webinar. So I'd like to start this presentation with a couple of notes. One is the goal of this presentation is to show you various techniques for prioritization of structural variations. And it is not to compare these methods in any way, but rather give you an introduction to them and how you can prioritize structural variants for identifying pathogenic things. So with that, I'll go ahead and start. We all know that there's thousands of genetic diseases around the world that affect millions of people. And there are several different types of testings that are predominantly existing. And here is a summary of these. As you can see, each one of these techniques has a specific target for structural variant identification and small variant identification. In case of Illumina sequencing, it's primarily designed for identifying single nucleotide variants and small variants that are below 50 base pairs in size with a very high precision. And you can see that the length of these sequences range from 50 base pairs to 250 base pairs on average. And the cost is about $1,000. This is with an error of approximately a couple $100 plus minus. Array such as microarray, agile, and comparative genomic hybridization is also predominantly existing. And this is for identifying copy number variation gains and losses that are typically greater than 30 kilobase pairs in size. And the main negative of this technique is that it misses on identification of any single nucleotide variations because it's a hybridization technique. Now, in more recent years, we have seen introduction of long-read sequences, such as Specific Biosciences and Oxford Nanopore technologies, both of which strive to sequence DNA in much longer lengths than the currently offered by short-read sequencing, such as Illumina, Complete Genomics, or Ion Torrent. So here, these two long-read sequencing technologies are still in development. They can, in theory, identify both single nucleotide variants and structural variations, but there's a bit of a caveat to these because the read lengths are quite below the repeat sizes that the human genome currently possesses. And the quality of these lengths of these sequences is not up to par with short-read sequencing at the moment. So in theory, yes, we can identify all different types of structural variations and small variations, but the quality of these is still quite low. And as you can see, the comparative costs for these long-read sequencing technologies is quite high compared to other technologies. And then lastly, there's optical mapping technology by Genomics, which has molecule lengths that are on average 300 kilobase pairs in size, and it's primarily geared towards identification of structural variations. And it cannot identify single nucleotide variants. And the size of structural variants is typically higher than 500 base pairs in size. And as you can see, the cost for optical mapping is $500 plus minus a couple $100, depending on whether you do it yourself or you give it to someone to map. And so the reason I introduced this slide is because going forward for genetic testing, it is very important to be able to identify different types of structural variations as well as small variations in integrated approach, so that when we try to identify the underlying genetic mechanism of the disease, we are able to find all the different types of variations. So in our institution, we're primarily focused on short-read sequencing provided by Illumina and optical mapping by Genomics. And the main reason for this is because with these two technologies, we can identify single nucleotide variations, small insertions and deletions, such as below 50 base pairs in size generally. And we can identify very large structural changes greater than 500 base pairs in size. That includes large deletions, duplications, copy number variant changes, inversions, and translocations. So I think many of the attendees are familiar with short-read sequencing technology and how it works. So in the next couple of slides, I would like to introduce the optical mapping so that everybody is on the same page. And so Optical mapping is a technique that utilizes DNA molecules that are megabase size, that are relabeled in specific sequence motifs, and these DNA molecules are imaged in nanochannel arrays to generate de novo genome assembly and perform structural variant calling against a reference of your choice, in this case, human genome. So the way the optical mapping works is we extract the high molecular weight DNA, and the user has a number of different ways to isolate this, either use what is provided by the company or use some other options. And so once the high molecular weight DNA is extracted, we use a direct labeling enzyme 1 that targets this CTTAAG locations throughout the genome, and it labels them with a green fluorescent dye. And then the backbone of DNA is stained with a different color, and then these molecules are pushed through the nanochannels provided. This is array of number of nanochannels that are in thousands and a couple of hundred thousands. And what you see here in the image is the staining of blue color of the DNA backbone, and then each one of these labeled sites is in green. And so the software automatically converts these images into text files that contain the size of the backbone and the locations of these green fluorescent lights labels. And so these molecules are put together in pairwise fashion until we assemble a consensus genome map that kind of looks like this. So all of the molecules are condensed into one, and then we know exactly where these locations or labels are. Now, when we are calling structural variants based on these against the reference, what we're typically looking for is any misalignment against the reference genome, either HT19 or 38. So here is an example of a deletion. So a number of labels are missing from assembled map in blue that were present in the reference genome. We can also see repeat expansions. So for example, here you see in the top middle that there's a lot more label sites are available. So there's a repeat expansion. We can see one half of the map aligning to one chromosome and the second half aligning to the second chromosome. And that's going to be a translocation. We can also see insertion, a tandem duplication, and inversions. So this workflow is typically what we use at Children's National. And I'd like to walk you through kind of the steps that we take to generate the genomic data and tools that we use to analyze them. So here with a data collection, we typically start with either blood. It could be fresh frozen, cells, also fresh or frozen, and tissue. We use the SP kit, which is provided by Genomics, to isolate high molecular weight DNA. And this DNA is used to perform both optical mapping and PCR-free library for short read sequencing on NovaSeq, on S4 flow cells to generate FASTQ files. And the same DNA, again, we use the direct labeling to perform and obtain optical mapping size, optical mapping data sets. For variant calling, we use basically the tools that are provided by Genomics for optical mapping. So it relies on Access, which we'll show a demo of in a few minutes, to perform the de novo assembly using BiNanoSolve bioinformatic toolset and call structural variations in the format of SMAPs. And as well for short read sequencing, we use either BaseSpace or Genox, which both are commercial companies, to perform short read alignment and variant calling to get the BCFs. Now for analysis, we really rely on 3 different annotation tools and packages to perform our variant annotation and identify the causative variants. So in case of structural variations, we will perform the variant annotation pipeline, which is again provided by Genomics through Access, and then we'll perform short read sequencing integration with a commercial company called Genox, again which I show a demo of, and then through Genox as well, we will use the VCF files generated by short read sequencing to obtain annotations for small variants below 50 base pairs in size, and perform variant classification. And so in this demo, again, I will start with going through access demo, and then we'll go to our own annotation tool with Nanotator, which provides an Excel output for all of the structural variations that we obtain from optical mapping. And then lastly, we'll go over the Genomics platform, which provides basically analysis tools for both structural variations and small variations with a comprehensive annotation toolset. So in part again, so we will examine the Binano Access, which is an end-to-end access platform that can handle data acquisition to variant annotation and filtration, along with visualization. We will discuss the Nanotator, an open source R package for annotation of structural variations developed at Children's National. And then we will walk through the Genomics platform, a subscription software that is capable of integrating optical mapping datasets and short-read sequencing datasets for a high-quality structural variant detection and annotation. So first, I'd like to introduce you to Binano Access. This is a web-based platform that is designed by Binano Genomics, and it offers an end-to-end management of optical mapping datasets. And I've listed a number of points here that we will go through as we go through the demo. So let me share my screen now. Okay, so as you can see here, this currently here is a Binano Access instance that is running on Children's National server. And when you log in, you're greeted with this image where you can access your project, experiments, and other settings. So in here, I'm going to show basically you for the data management part. You can click on experiments, and this tab is basically experiments that are connected to Sapphire instrument that you're able to run and obtain genomic data. So if we look, for example, one of our runs that we've previously done. So let me just open that in a new tab. You can see that how much DNA is being generated for each of these different runs in each of the chip flow cells, and the corresponding map rates and statistics. Once the run is completed, we can go to projects menu. And as you can see, now we have in here, I have a number of different projects. So we're going to click one of them. And you can see that, one second. And you can see that we have a number of samples present in this project for which we have the molecule files. So if we can filter this by molecules, you can see these are all the different samples that we have obtained the molecule data for. And now we're able to use these BNX files to perform de novo genome assembly and variant calling. And that's performed here. So we have a number of options to perform. Again, I'm not going to go through all of these, just the main ones that we use currently for our analysis. And that's called de novo genome assembly. And what this assembly does, it puts all the molecules together to generate consensus maps and compares to the reference genome to identify structural variations. And we have done that for a number of samples here, assembly. And you can see that all of these samples have been assembled, and we have variant calling information. And so once we perform this, Binano Genomics through Access also offers annotation. So if we choose a particular sample here, for example, sample 1, we can see that for each assembly, we have performed variant annotation. And what variant annotation does is it takes all of the molecules, for example, in a trio, if you have parents, and it compares the molecules to the proband's assembly to make sure if the variant is actually present in any of the parents. And it also provides gene overlap information for structural variations, as well as providing frequency information in the Binano Genomics database. So we're going to take a look at one of the analyses here. But before that, I want to show you one example where when we have assembled the genome, we have two different ways of looking at it. So one, here's an option to look at the whole genome copy number variant calls. And so if we open it in a tab, we can see kind of a similar to array type of representation. So on the bottom, we have all the chromosomes, and then we have the copy number variant on the left side. So if we zoom in, let's say on chromosome 1, we can see that most of the chromosome 1 is diploid, so 2 copies present. And if at any point there is an alteration, here's an example of that. For example, chromosome X is present at a higher number than 2 copies. We're able to detect that by showing this gain of chromosome X. And then the second view, again, if we click here, it's going to be maps to references with structural variations. And what this menu brings up is a circus plot with a summary of all of the structural variations that have been identified in your sample. And we can do a number of different things with this. So there's a filtration tab here that we click, brings up a new window. And what this window does is gives you a user an option to select different types of filtration criteria. For example, for insertions and deletions, we can choose recommended quality scores by non-genomics. We can set thresholds for size of these things, including the duplication. And we can also look at variant annotation filters that are provided with trio analysis. For example, we can check whether we want all of our variations to be less than a certain percentage in the reference database. In this case, this is the reference database provided by Genomics. And we can also choose whether we want the structural variants to be present in the parent assembly. So for example, if we click this one, this is quite important for identifying de novo variations. We can select either all structural variants or structural variants not found in parent assemblies. So if we select this option and we click apply, we only see structural variations that are de novo. In this case, there's only a handful number of structural variants that are classified as de novo. And so I'd like to actually explain what the circus plot different levels are. So here on the top, what you see is a chromosomal cytogenetics banding, G banding. Right below that is this box is representing the number of structural variants that are located within the chromosome. And then below that is the copy number variation for each chromosome. So for example, in this case, if you look at chromosomes 23 and 24, which are X and Y, respectively, you will see that chromosome 23 is at level 2x and chromosome Y is 0. So this case is 46XX because there is no other structural variations present. And so if we want to look at any of these variants, we can simply navigate to them and click them. And what this will bring up is the visualization of that variant in a linear view. So we will have here, give it a second. So we will have a table browser on the bottom here, which shows us different variations that are present on chromosome 16 in this case. And one second, let's just click this. I'm sorry, I have to apologize because I'm connected to our network at VPN, so that could make things a little slower. So if we're interested in looking at some variants, let me orient you here as well. So here we can see chromosome number 17 as reference. So these are in silico digested reference genome. In this case, this is HG19. And what we see is two different maps that are aligning to the reference genome 17. And we can see these maps showing structural variations. For example, if I zoom into this region that shows a deletion, it will automatically point the map that is generating this to call a deletion. In this case, you can see that all of these labels in the middle of the reference genome are missing. So that indicates that the sample map here has a deletion. Okay. We can also there's a number of different things also possible to do with access. I don't want in any way to go over all of the different features. If you're interested, there is a very nice white paper available on the website that you can take a look at these. But what these filtration criteria, I just want to make a comment here, are allowing the users to do is to basically select for all of the rare variants that could be possibly pathogenic depending on the disease type. And it also shows in the table below here the genes that are overlapping this particular structural variant. For example, if we also look at the canonical genes, so in this case, you can see that this gene is likely being deleted in our sample. Okay. All right. So next, I would like to go, if you look at the slides again, I would like to go over an annotation tool that does a little bit more in terms of annotating the structural variations produced by optical mapping. And this tool has been developed at Children's National by Dr. Srajit Bhattacharya. It's available, it's open source, it's available through GitHub and by a conductor. So one of the things that we wanted to have in the annotations is classification of different types of structural variants. And here is an example. So in the top, you can see that there is a reference genome. And on the middle, we say, if there is an upstream deletion of gene X, we would like to get information. So in the table below, you'll see the deletion, the overlapping gene, there is nothing. Gene X upstream, there is a deletion. We have the orientation of the gene expression. So in this case, it's gene X is on the positive strand, and we have the distance away from the structural variant of that gene. So it's an indication that the deletion is upstream, and it could have an impact on expression. Okay, so next example is a insertion on gene Y, and you can see it overlaps with gene Y that is transcribed from the negative strand, and it impacts 20% of the gene Y, overlaps 20%. And in addition to this, if the user has any RNA sequencing expression data or any type of expression data, it doesn't have to be RNA sequencing, we are able to take the expression values for these genes and paste them in a separate column next to these values, and to see whether any of these structural variations have an impact on gene expression. And so here we also provide several additional things, and primarily that is we recalculate the structural variant frequency in a number of different databases, including by Nanogenomics database, DGV, Database of Genomic Variations, and Decipher. And the main reason for this is that one, we want to see the frequency in more than one database. And we also want to see what is the frequency in our internal database. So if you have more than 10 samples, we can merge the annotator output, merges these data sets into one big file, from which we can estimate the frequency of the variant that you're studying. So here I'd like to show a demo here. Let me just minimize this one. So what the annotator does is very simple in a sense, because we take the SMAP outputs or variation outputs from by Nanogenomics, and what we do with it is we provide additional fields. So for example, here I've highlighted a number of different interesting criteria that are present in our Excel format that are used for users to filter things. So here, when the variants are classified, they're put into different categories by inheritance. In case where you have the inheritance of parents, if you don't have any parental samples available, we can put these in the same categories. For example, insertions, deletions, inversions, and translocations. Or if you have a control sample, only one of the parents, for example, is available, then we'll have shared and not shared variations with either of the parents. Or in case of tumor, whether it's present in the tumor sample or not present. So here you can see that we divide and categorize variants based on inheritance patterns of this trio sample. For example, it's insertions, deletions, duplications that are de novo, so not present in parents. Insertions, deletions, duplications that are present in both parents. So this tab here is primarily used to identify recessive variations. So for example, a variant is present in a homozygous state in a patient, but it's both parents are heterozygous. We also have insertions, deletions, duplications to be set as compound heterozygote. For example, if two structural variations are within approximately 1 megabase of one another, then they will be identified and put in this category. Currently, none of these, so what I want you to pay attention is that none of these are filtered yet. So this is the raw output. And the next file I'll go over, it will have all the filtration. You'll see how it's powerful to decrease the number of variants that you need to look at manually. The next one is inversions, then translocations. This particular case did not have any translocations. And then insertions, deletions, duplications that are inherited either by mother or by father. And then last tab, typically here, we have primary gene list overlap. So all of the previous tabs also have primary gene list overlaps. So in this case, when a user provides a gene list of interest, we will use that gene list to overlap them with structural variations of interest. And we will also provide a term list. So for example, if the terms that are overlapping, the genes that are overlapping with structural variations have a phenotypic term that is present within the proband, then that will be also provided. And then lastly, we just print all of the variations. So if the user wants to take a look at all of the variants that have been identified, they're listed here. And so when we do our filtration, for example, we filter by quality scores, we filter by frequency, we filter by the different types of categories, we can see that we typically decrease the number of variations that we need to the user needs to look at. And so this greatly reduces the amount of time that someone needs to look at these variations and analyze them. So in this case, we have an example of developmental delay, which we have filtered down to de novo variants, and we have several tabs that I'd like to go over. So here we have the gene list that are overlapping the structural variations. In this case, if we look on the top, all of these genes are overlapping this. And if you look individually at all of these genes, we can see that whichever gene is transcribed on which strand and how much of the gene is being overlapped with the structural variation. We can also see which genes are upstream and downstream of these of the structural variation. We can see the distance in kilobase pairs, and again the orientation of the transcription of the gene. After that, we have some frequency information in this DGV. Internal frequency, how many times this variant is seen in your own cohort, and the mother and father zygosity. So in this case, because it's de novo, we don't see any of these come up. And then lastly, what I would like to show is another case where we have RNA sequencing information, for example, here, where we had RNA information for all of the trio members. You can see that we look at the overlap gene here, which is EHBP1. And we also are able to see the gene expression in different trio members. So in proband, it's 15 transcripts per million reads. And in parents, it's variable, so 10 and 20. So what this really allows a user to do is to use a simple Excel format file to easily filter variations based on these key metrics: frequency, gene overlaps, primary gene lists of interest, and categorize the variants based on inheritance. So if there's a specific inheritance mode that you'd like to study, it's available through all of these tabs. And then lastly, there is a results tab for where you're able to, in this case, all of these tabs have their corresponding naming. So insert here is de novo, homozygous, compound heterozygous, etc. And if you end up finding a structural variant that you think is worthwhile, you just paste that here, and you have a nice summary of findings. Okay. So next, I would like to go over this integration, and the primary reason for this integration tool, again, as I mentioned in the earlier slides, is that whenever we're doing our structural variation analysis, we always prefer to do them with utilization of short reads. And the reason for this is, of course, we can look at structural variations, but if we're not able to see the smaller variations that can have a major impact, depending if they're in the exon of a gene that is pathogenic, given the disease that you're studying, then we want to be able to detect that. And with both Access and Annotator at the moment, we're not able to do that. So there's really, we partnered a couple of years ago with both Genomics and Genomes to basically be able to do that kind of analysis. Web user interface. And so the way this works is we provide this output of optical mapping to Genomes. And what they do is they align the short reads with a graph alignment based on the structural variation breakpoints. And they do a SV calling based on those alignments. And what this joint SV calling allows to do is to basically greatly increase the quality of structural structural variations identified by short read sequencing, because we are guiding kind of the alignment of these with optical mapping data. And it also helps greatly to narrow down the structural variation breakpoint identified by optical mapping. So let me just start the demo now. So I'd like to start with a demo here on the, again, the Genome's highly comprehensive. And so if I were to go through all of the functions and the ways to filter data, it would take a very long time. So I really want to introduce the structural variation analysis as well as integration with short read analysis in this talk. So what we're able to see is an example. So this is EGR001, is the same sample as I was showing in Access and in Annotator. What we see here is number of the phenotypes that we have for the patient that we entered, and we have number of filtration criteria here on the left. Again, I'm not going to go through all of them, but the most important ones are, again, the frequency, number of times it occurs, and the variant colors. So Genomics actually what does is using number of different types of structural variant calling algorithms on top of binano. So for example, if we want to select only for binano variants, all we need to do is click for binano variations. And this list here will automatically change and show only variations that were identified by binano and have a better breakpoint resolution. So for example, we can, if we want to identify more information on this, we will just click on that tab and we will see the start and end breakpoints and the tool that was used to identify this particular structural variations. We'll have the range for the confidence interval for start and end. Additionally, if the reason that Genomes is quite good for annotation is because the ACMG guideline-based classification of structural variants as well as small variants is built in. So for example, if we want to take a look at pathogenic and likely pathogenic variations, we're able to do that. And what we see here is an example of a likely pathogenic variant with a very high confidence. And what we can do is we can click on that variation. And then we have, again, we have a lot of different types of annotations that we can use to classify this variant. One of them is ACMG guideline classification developed by Genome. So these are all ACMG guidelines for structural variations. In this case, it says uncertain. We can look at the assessment tool. So we have a little browser here that shows different structural variations have been submitted by public databases. So in this case, if we hover over it, we can see that the CleanVar has classified as VUS. The Cypher has this variant classified as pathogenic, etc. A number of occurrences in different types of databases. For example, here in DGV, this variant has not been seen. In NOMAD database, which has thousands of samples, has not been seen. We can also take a look at our internal database. So again, as you upload your samples into the Genoox platform, you're able to classify variations that are present in all of your samples. So if any other sample had within this platform this particular variation, that will be listed here. And we can see families like us, for example, if parents of this case also had it. And then lastly, there is an option to look at actually alignments of short reads. This is by IGV. And if you want to take a look at the short read alignments, it's available here. Additionally, we can take a look at any of the keywords that we have entered. For example, the keywords for this case were all of these listed here. And we can look at if this gene has any associations with any of those terms. So for example, this gene has an autism association, and we can click it, it will take us to that page. And we can also look at publications that are present here. So anytime this gene came up, it's an easy access to these publications. Okay. So one additional thing I'd like to show is actually a new feature that was developed recently in the last few months. Is this tab? I think I already have it open here. So this compound single nucleotide variation and structural variation. So what this allows you to do is instead of manually, when we used to do analysis of structural variations and analysis of short reads, and then seeing if there's a potential of compound heterozygous variant present. So what this basically does is an automatic way of finding a gene that has an overlapping structural variation, in this case that, and a single nucleotide variation or a splice variation in this case. And so what we're able to do again is classify them by ACMG guidelines, whether they're likely pathogenic and pathogenic, or we can choose whatever we want from this drop-down list. And then we can click gene name and basically go inside, look at the single nucleotide variant, internal frequency, and etc. Okay. So I'd like to end the demo here and follow with a couple of summary slides and get to the questions. So I've listed a number of pros and cons for each of these methods. One thing I'll say is that we currently use all three. And the point of this presentation is not really to suggest which one is the best tool to use. In reality, all of them have functionalities that are unique, and then they also have a lot of functionality that is in common between them. And so again, I'm not trying to compare these. These are just really thoughts that we came up that kind of show why each tool is good compared to some of the other tools that are available. So Bionano Access is again, this is a free-to-use software that is provided by Bionano Genomics. It has a professional support. You're able to store all of your optical mapping data in this platform. You're able to manage it, do analysis, visualize. So it's a very comprehensive end-to-end tool. Some of the cons are that if you have any gene expression data, there's no way to integrate it. It is RAM intensive. So for example, if you're working with a laptop and you open several tabs on your web browser to look at different samples simultaneously, it will become very RAM intensive and it might be very slow. And some of the structural variant frequency information is automatically listed for Bionano Genomics database. For Notator, we have it's a free software that is open source. We provide a number of different frequency information, including the internal database. We can do phenotype-based gene list filtering, gene expression overlaps, variant categorization based on inheritance, and it's a very fast and simple. So there's no web user interface. There's no need to click things. Once you have your set guidelines, you just implement them, and you have the variations present. So it's very easy and fast to take a look at all of the variants that you're looking at. And then so some cons. So if somebody is a web user, likes to use the web user interface, it's not available. It does not have the professional support. So if you're facing an issue, then the likelihood of having that addressed is a lot less than with a professional support. There is no visualization. So anytime we currently find a structural variant in using the annotator, we always go back to the bind on access to generate some visualization images. There is no VCF support currently, but this is coming soon. We're going to add the nomad support for structural variations and support for the exon information. So example, which exons from the genes that are overlapping structural variants have been deleted. And then lastly, it's Genox. Genox is a professional annotation software. It has support, web user interface, ton of annotation information, including the publications, the classification based on ACMG guidelines. It provides integrated analysis of short reads with optical mapping data. It has a sequence browser, so to visualize short reads and reporting. So again, some of the cons included it's subscription-based, so you'll have to sign up for some sort of a paid mechanism. Currently there is no support for gene expression overlaps. It's compared to an annotator, both Access and Genox are quite slow in terms of filtering variants. So every time you change something on the filtration, you're going to have to wait about, depending on your internet connection and sites, anywhere from a few seconds to up to 10 seconds. And it currently does not have the same visualization support as does the binary access. So I'd like to end with a summary of the way that we're thinking about identifying genomic variations. And here we have three different types. So we try to use the optical mapping because of the length of the molecules that we're able to get to identify structural variations and copy number variations. We pair that information with short read sequencing in this case for identification of smaller variations that are below 50 base pairs. And of course, when we do that integration with optical mapping and short read sequencing, there's going to be some variations between 50 and 500 base pairs that we're going to miss. And so for those, depending on how fast the long read sequencing is going to catch up with quality and price, that it could replace the short read sequencing. But that's up to discussion. And then lastly, not for all samples, but more recently, we would prefer to perform expression analysis in these patients as well, just to see how some genomic variations that are in splice locations affect the gene mutation, as well as if there is any non-coding variations, how does that impact the gene expression. So with that, I'd like to thank the laboratory. I'd like to thank all of the lab members who have helped with making the annotator data analysis. I would also like to thank the Binary Genomics support team and Genome support team. They have been very responsive, very helpful for all of the questions that we've ever had. So with this, I'd like to end and open the floor for questions and answers. Thank you.